The recording started. Okay, let me share my screen. So, is my screen visible to all of you?
Yes, sir.
Okay, thank you for confirming. So, good evening, everyone. Myself, Karan Kumar. I am research scholar at IIT Guwahati, and welcome to this second live session on fundamentals of protein chemistry. So, today we will be going to discuss about the week 2 assignment. These questions that I will be taking here is not related to the current assignment that are in the week 2 that you might have gone through, or some of you might have submitted it. But I will be discussing the previous year's question and some of my sample questions that will help you to solve the current questions. And also, I will tell you the techniques how to approach the questions and why these questions are important for your upcoming exams, which will help you in your in semester examinations and also in your various competitive examinations related to the protein chemistry or protein biochemistry related to your biotechnology field. So, as I have already mentioned that, and you might be aware that this course is listed in the SWAYAM NPTEL portal under the and the under the guidance of Professor Swagata Dasgupta. So I am your TA for this course, and I will be taking the weekly live sessions. In those, I will be discussing the problems, sample problems, and also will tell you how to approach these problems. So let's start this meeting, and I hope this will be beneficial for you. So please confirm whether my screen is visible or not.
Yes, sir.
Is
It
my
is
screen
visible.
visible? Okay.
Yes, sir.
Thank you for confirming. Let's start the meeting. So the concepts that were covered in the week 2 are levels of protein structure, torsional angles, rotamers, amino acid sequencing methods, types of interaction, protein folding, and various type of protein quaternary structure and their forms. The important keywords that you might have come across that are 3D conformation, amino acid sequence, and terminal amino acids, C-terminal, torsional angles, mass to charge, peptide backbone, planarity of this backbone, then polar and non-polar surfaces, binary matrix, then contact plots, various terms related to the protein architecture. So I hope you all have gone through the lectures on week 2. And also I would like to tell you that I took the session for week 1 also. If you have not attended that session, you can see my live recording of the live session in my YouTube channel. I will share I will show you which is the YouTube channel. So if you have not subscribed the channel, you can subscribe it and watch the videos related to the previous assignment such as week 0 and week 1. So the channel name is NPTEL-PMRF TA NOC22-BT62, which is the course code. And also you alternatively, if you go through the SWAYAM portal of this course, then there is a section added here, which is problem solving sessions. So all the lectures that I will be taking, that will be listed in this problem solving sessions. So, and if you click here, the Excel sheet with prompt, and here you will find the link, or alternatively, you can also go through my YouTube channel for this. So let's start it for the week 2. Before starting, as this is a live interaction session, so I would like to interact and I would like to get your opinion on the week 2 module, like module 2. Here, a few general questions before starting this problem solving session. I would like to ask you, what do you understand by the term protein architecture? You might have gone through the module 2 lectures from lecture 6 to lecture 10. Can someone unmute yourself and answer this question? You can answer in any language you are comfortable, either Hindi or English. So language is not a barrier, but yeah, I will try to make this session as interactive as possible. So please answer this question. What do you understand by the protein architecture? Can someone unmute
Can
themselves and answer
can I
this?
answer, sir?
Yes, sure, sure.
Okay.
Yeah, go ahead. What is protein
So,
architecture?
so protein architecture means the arrangement of the different amino acids to the peptide bond in terms of the primary structure or in terms of the secondary structure having the alpha helix or beta sheet involving the hydrogen bonding, or they can also arrange themselves in the tertiary structure by folding by certain loops and turns, and including the quaternary structure which is there in hemoglobin, the four subunits which are interconnected by the non-covalent interaction. So that is, according to me, is a protein architecture.
Okay, very good, Karan. Uh, Anyone else would like to answer this question? They can unmute themselves. Anyone? Okay. So as rightly pointed out by Karan, that protein architecture is nothing but to study about how they are arranged and how they are folded and what are the chemistry behind it. Not only not only talking about the primary structure, not only talking about the secondary structure, because the functional form of protein is either in the tertiary structure or quaternary structure. So as studying the protein architecture is much crucial to know behind how, because all the proteins are nothing but they play all the roles in our human body or any organism that exist in planet exist on planet, like they ranging from enzymes to membrane protein to anything else, whatever you come across, like hair or your bones. Like in your bones also there are some proteins, then in joints. So proteins play a various role in repairing, in maintenance, and then in playing the functional roles. So that is why if you want to know how this particular reaction is being carried forwards by a certain enzyme, or if certain repair mechanism is happening in our body or in any organism, if certain phenotypic behavior is being showed by that organism, then the role behind it is the protein, like the main key player is the protein. So to study their function, to study their interaction, to study their characteristic, you need to know the protein architecture. So then that also answers my second question, that is what is under why understanding of protein architecture is so much important in protein biochemistry or protein chemistry, whatever you call this. So, and throughout this session, I would also, in the end of this lecture, I will also tell you the scope of this topic with research perspective, why protein architecture is important as a research for research people. Like, and there is another stream or subject which is related to this terminology, which is structural biology. So in that also you study the protein architecture and basic fundamental behind this. So, and that is the research perspective of that. Apart from it, can someone specifically mention what can be the research perspective of studying this topic? Can somebody answer? Unmute themselves, unmute yourself, and then you can go ahead. Anyone? Okay, no problem. So we will start the question answer session. This was the brief introduction about the protein architecture and the module 2. So let me give you a question, a set of question that were in like that were there in the previous year's assignment. So your first question is, I have modified this question a bit. So your first question is, which of the statement is or is are correct about the protein in non-aqueous solvent? So your option A is placement of hydrophobic amino acid residue within the interior of protein. Option B is placement of polar amino acid residue around the exterior of protein. Option C is placement of hydrophobic amino acid residue around the exterior of protein. And option D is minimization of entropy by the formation of water solvent cell around the protein. So what are your understanding from this question and what can be the correct answer? Any guess? Or if somebody know about this question, they can answer this question. Unmute yourself and answer the first question. Which of the statement are correct about protein in non-aqueous solvent? What could be the answer?
Sir, option A can be the answer because in non-aqueous solvent we are not having a water, so it means there can be a polar solvent, or you can say there can be organic solvent. So the hydrophobic part, which is the organic part, must be within the interior of the protein.
Okay. Any other? Any other answer is there? Please note this is non-aqueous solvent. Okay, we will come to this question later. The second question is, quaternary structure is possible for a protein only when option A, the amino acid cysteine is present. Option B, all amino acids have non-polar R groups. Option C, two or more polypeptide chains are present. Option D, a protein chain bends back upon itself. What could be the answer? Correct answer for this question?
Sir, option C, two or more polypeptide chains are present because in hemoglobin there are 4 subunits. So according to me, there must be 2 or more polypeptide chain for the quaternary structure.
Good. Okay. Question number 3. Which of the following does not affect the stability of an alpha helix? Which of the following does not affect the stability of an alpha helix? Option A, hydrogen bonding. Option B, bulkiness. Option C, interaction between R groups spaced 3 residues apart. Option D, occurrence of alanine and serine residue. What could be the correct answer for question number 3?
Sir, option D, occurrence of alanine and serine residue, because hydrogen
Okay.
bonding is also important for alpha helix, and bulkiness of the side chain also play an important role, as well as the interaction of the R group also play an important role for deciding the stability of the alpha helix. So it must be option D.
Okay. Okay. Are there any other answer? If somebody want to answer the question number 1, 2, 3, they can unmute themselves and put their view, like they can tell why they think about particular answer. Is there someone who want to answer the question number 1, 2, 3? Okay, so let me discuss these 3 questions. Question number 1 says about the non-aqueous solvent. The protein is submerged in non-aqueous solvent. Then what will be its fate? Like the different group, how may they appear as their solvent around them is not water, non-aqueous. Non-aqueous, as you rightly said, that this may be the organic solvent or something else, but non-water. Non-aqueous means non-water. So normally what happens, like the hydrophilic amino acid residues. So hydrophilic amino acid residues normally found on the out on the exterior region of the protein in aqueous solvent. Okay, why? Because when hydrophobic amino acid residues are present on the outer region of aqueous solvent, then there might be the interaction which makes the structure in not folded in proper manner, or also it creates uh, not it it destabilizes the structure. So because the hydrophobic amino acid residues, they don't interact with water, that is why in aqueous solvent they are normally present in the interior of protein. But here the question was in the non-aqueous solvent. So in non-aqueous solvent, the hydrophilic amino acid residues will be buried inside the core, and the hydrophobic amino acid residue will be exposed on the exterior of the protein. They might be exposed, but mostly because in the crystal, like normally they can be or they may not be, because if crystal water is present in the waters, some water molecules are present inside the protein, then they will interact with the hydrophobic that can destabilize the protein. But in the proper non-aqueous solvent, the hydrophobic amino acid residue will be present on the exterior of the protein. Okay, so that there will be a hydrophobic interaction, and that will stabilize the protein structure more accurately, other than the if the hydrophilic amino acid residue is present in the surface. So the correct option for question number 1 is C, the placement of hydrophobic amino acid residue around the exterior of protein. And second question, talking about the second question, the quaternary structure is possible for a protein only when because as already as rightly mentioned by some of you, I cannot see the name who answered the question. Yeah, but he rightly answered this, that quaternary structure are the result of when two or more polypeptide chains interact and they form a certain kind of interaction, weak interaction, or certain kind of interaction such as disulfide bridges or any other means. So, such as the hemoglobin have the four different tertiary structure attached to it. So that is the structure of alpha 2 beta 2. So that is why the quaternary structure itself, the name says that there will be involvement of two or more polypeptide chains. So correct answer is C. And question number 3, which does not affect the stability of alpha helix. Here, there is no role of alanine and serine to destabilize the alpha helix, because the alpha helix breakers are glycine and proline. Glycine uh, because of its smaller in size, and proline because it has imino group. So because of that, they usually destabilize the structure. Other than that, all amino acid can be there in the alpha helix, and other options such as hydrogen bonding, bulkiness, interaction between R group spaced 3 residues apart. These all kind of answer are the right one, because all of them affect the stability of alpha helix except the occurrence of alanine and serine residue, which is the correct answer. So option number D. So I will give you a brief about the protein bond formation and the angles such as dihedral angles. You might have come across this in the lecture. So let me tell you, and this will be useful for your upcoming questions. So here, when two amino acids, when two amino acid residues are here, when two amino acid residues such as one amino acid 1 and amino acid 2 are joined to form a peptide bond, which is CONH bond. Here is the peptide bond. So normally the convention is you put a N terminal of the first amino acid in the left hand side, while the in right you will find the carboxylic acid terminal. You also call it C terminus and N terminus. So, and during the condensation process, normally the two amino acids or polypeptide chain formation is happening through the process named as condensation. Okay. So through the condensation process, one water molecule is released, and then this bond is attached to nitrogen, and then you form a peptide bond. Okay. So the geometry of it, like the nitrogen has a lone pair, right? So this lone pair, uh, this lone pair contribute to the conjugation of this pi electron delocalization, and because of that, this peptide bond, which is between the carbonyl carbon and NH of the second amino acid, they in a kind of uh, partial double bond character, which is characterized as a 40% double bond character. Okay, due to the pi electron delocalization. Okay, so this lone pair will come here, and then this double bond will go here, and the on oxygen you will find a negative charge. This will convert as a single bond. So then this peptide, due to this peptide bond, this feature, because of the delocalization of electron, this peptide bond formed, this peptide bond called as a planar. This give a nature of the planarity to this peptide backbone. Okay, so you don't see the rotamers, the like around it, because this is a kind of partial double bond character. So the rotation is not possible around this bond, peptide bond, but rotation can be there in between the two other bond. Here, if you identify here, the peptide bond is this one, right? The peptide bond is this one between the NC. Okay. Oh, sorry. Yeah, this is also a peptide bond, and this is also a peptide bond. Okay, so if you find a angle, because this N C alpha C beta and O, this form a two plane. There are two planes involved in that, and in between the two planes, the the rotation can happen with due to these single bonds, which is C alpha C and NC alpha. So NC alpha bond is termed as the phi angle. However, C alpha and C bond is called as a psi angle. So normally you see a rotation of these two around these two bonds, and which is also being plotted, and that plot is called Ramachandran plot. Okay. These all things I am telling you to just recalling all the concepts that have been discussed between the in the protein architecture module. So, and you can have the other rotations around the side chains. In the side chains, there will be the different single bonds will be present, and then those dihedral angle will be termed as the chi. Okay, the terminal as per UPEC, tell you name them as a chi, and other dihedral angles that are present in the peptide backbone, that are termed as the phi, psi, and omega. Omega is around the peptide bond. Okay, which is represented here. Okay, so here peptide bond is omega, then C alpha psi bond is called psi. So you can remember that by CC psi, and NC alpha is phi. Okay, so there will be a restricted rotation about the pi bond, and due to the partial double bond character, it is a rigid planar structure. Okay, and as a consequence of resonance interaction of the pi electrons over the four atoms, which is NCO, three atoms. So now coming to your next concept, and due to this, one thing I missed here, and if because there is a partial double bond character, so this also have a single bond character, and there will be a restricted rotation, which results you as a trans and cis form of the peptide bond. you see a very few interaction between them, and the normally existing, or you normally don't see the cis conformation except in proline. Proline amino acid, you only see the cis conformation, and other than that, you don't see the cis conformation more prominently. Now coming to your next question. An alpha helix is formed by hydrogen bonding between the hydrogen of an amide group and the backbone carbonyl carbon. How many amino acids upstream of it? So, can someone answer? This question is related to the stability of alpha helix and the formation of alpha helix. So, can someone unmute and answer this question? An alpha helix is formed by hydrogen bonding between hydrogen of an amine group and the backbone carbonyl carbon. How many amino acid upstream of it? Option A 3, option B 2, option C 5, option D 6, option E 4. What is the correct answer? Anyone? Okay, the correct answer is E, which is 4, because you must have until unless it is mentioned. Normally, alpha helix present in the body or in normal in nature, that are the right-handed alpha helix. So in the right-handed alpha helix, you see I to I plus 4 hydrogen bonding, like the hydrogen bonding formation between I and I plus 4 amino acid residue. So if you have in alpha helix, like here in a full turn, okay, let us say this is a full term as per the helical wheel structure here. So the amino acid residue which are present here at let term as 1, then 2, then 3. Likewise, if you term certain amino acid which is ith, okay, so it in right-handed alpha helix, this ith amino acid residue will form a hydrogen bond with I plus 4th, means every 4 amino acid residue will interact with this ith. Like the first interaction will be 1 and 4. 1 and I plus 4 means if I is equal to 1, then I plus 4 will be your I plus 4 will be 5, right? So you will see the first interaction between 1 and 5, first and fifth amino acid residue, then second and seventh amino acid residue, second and sixth amino acid residue. Okay, so likewise, so therefore the correct answer is 4. Okay, so explanation is given here. Alpha helices are formed by the hydrogen bonding involved an alpha carbon bound amine group, hydrogen and carbonyl group attached to the amino acid of 4 amino acid residue upstream. Okay, normally the direction of alpha helix is upside. So either in right-handed alpha helix or left-handed alpha helix, you will see the in the upstream, the I and I plus 4 amino acid residue will interact and they will form the hydrogen bond, and that stabilizes the alpha helix structure. Okay, so your second question is, which of the following best describe a characteristic of protein motif? So your first option is a commonly occurring arrangement made up of tertiary structure. Option B is a commonly occurring arrangement made up of multiple secondary structure. A unique arrangement made up of secondary structure found only a single protein. And option D, a unique arrangement made up of tertiary structure found only in a single protein. So what do you understand from this question? Before I would like to suggest, in any of the exam, if you attempting a question, first try to understand what the question asks and what are the keywords present in the question itself. Like in this question, it is talking about the protein motif. So if you have, ma'am has already discussed about the protein motif in lecture number 10. So I have included this question. This was not present. This question was not present in the current assignment or the previous assignment. So I have included this question in my slide. So try to understand this question and answer it. Which of the following best describe a characteristic of a protein motif? Okay, and can anyone unmute themselves and answer? Anyone? Okay. So you know the protein motif, or you also call it as a super secondary structure. Super secondary structure means that are not alpha helix or beta sheet. They might be the combination of it, or this protein motif or super secondary structure, they does not have any kind of proper conformation that are repeating itself. Like in alpha helix, you know that the amino acid where the hydrogen bond will form between the 1 and 4th amino acid. In beta sheets, either if they are the parallel or anti-parallel, then there will be a bond formation between certain there will be plating, they will be forming a particular kind of interactions only. But in secondary super secondary structure or protein motif, such as turn, loop, or other conformation which are not generalized, so those are called the protein motifs. So these are called the defined arrangement of the secondary structures within the protein, and it is commonly occurring enough to have the identified structure. An example would be the beta alpha beta loop. Okay, beta alpha beta loop is kind of uh, a loop where you see the both of the characteristic in between, like that is joining the alpha and beta beta secondary structure. So in that, so while the arrangement is made up of the secondary structure, the overall motif itself can be considered as the super secondary structure. Okay, or possibly even the tertiary. Okay, through it components, the second components are, though its components are secondary structure. So motifs do not necessarily have a defined function across the different protein or protein domains on the other hand do. So a hint is given here. So can now somebody answer the question based on this definition or the hint? Which of the following best describe a characteristic of protein motif? Based on this description, I find the option number B, which is a commonly occurring arrangement made up of the multiple secondary structure termed as the protein motif. So correct answer is B. Okay. So coming to the next question here. So this is very easy question. I hope you will be able to answer it. So in the above image ABC, choose the correct structure of the protein respectively. The chains are colored coded. Okay. So now, can someone attempt this question of question number 4? What
Yes,
is
sir.
A, what is B, and what is C? Yeah.
Sir, A is a secondary structure, B is a
Okay.
tertiary, and C is the quaternary structure.
Okay. Very good. Rightly identified. So A is alpha helix here, which is a secondary structure. Then this is the combination of alpha helix and turns. So it is kind of a tertiary structure. And here you see two or more polypeptide chains are folded and then they are interacting. So this is the example of quaternary structure. Okay. So now your question number 5 is glycine and proline are the most abundant amino acid in the structure of. So to attempt this question, you must know the importance of or the characteristic of glycine and proline in protein sequence or the in the level of protein structure. So your options are hemoglobin, collagen, myoglobin, and insulin. So in which of this structure the glycine and proline will be abundant? Can someone answer?
Sir, option B, collagen.
Option B, collagen. Why do you think so? Why do you think so?
So it can be due to the like a muscle protein, something related to myosin. So that is why proline is an amino acid and glycine is the simplest amino acid which is without having any chiral carbon.
Okay. So what does it have to do with the glycine property that it will be in the collagen? So the correct explanation you can give it as like the your answer is correct, which is glycine and proline will be abundant in collagen because if if the repeating amino acids are glycine and proline, then that particular kind of protein will not be forming the secondary structure such as alpha helix. Like it cannot form the alpha helix because both of these amino acids are the alpha helix breaker. Either they will be found in the parallel beta sheet or anti-parallel beta sheet. So only the collagen here have the beta like because this collagen is a pleated sheet is it is a rigid structure and it does not have the coil structure. So like hemoglobin and myoglobin or insulin, all of them has the quaternary structure which involves the secondary alpha helix conformation and other. But if the glycine and proline are repeating in a particular protein sequence, then the because this will interrupt in the formation of alpha helix. So that is why the correct option here is the collagen because collagens are the straight beta pleated sheets. Okay, and these are the fibrous protein. And other than the other options are the globular protein. Okay, so that is why the correct answer is B. So let me discuss. You might be aware that primary structure are the sequence of amino acid. Then these form a beta sheet, which can be the parallel or anti-parallel fashion. Then you will have another secondary structure, which is alpha helix. Then when these two forms are folded and the protein is folded in its native form, then it is called the tertiary structure. And when two or more polypeptide chain interact with each other and then they form the globular kind of protein, this form is called the quaternary structure. There are two type of alpha helices, which are left-handed alpha helix and right-handed alpha helix. In both the direction will be in upstream direction, and you can imagine this by the rising. If you curve your fingers, right hand fingers, then it will form a kind of a right-handed alpha helix. And if you fold your left hands, if you curl the finger of your left hand, then it will form a left-handed alpha helix. Other than that, you also see the alpha helix such as pi helix, then 310 helix. So these all where the alpha, right-handed alpha helix, left-handed alpha helix, 310 helix, pi helix, these all varies in in terms of the formation of hydrogen bonds between which number of amino acids interact with it. So normally, as I have already told you, unless it is mentioned, the major type of secondary structural component that are present in human body are right-handed alpha helix, and they form I to I plus 4 hydrogen bonding. However, in 310 and pi helix, they form in 310, they form I to I plus 3, and pi helix, you see a hydrogen bond formation between I to I plus 5. So the important points to remember in this context is the most commonly found helix is right-handed alpha helix. Then hydrogen bonding pattern is I to I plus 4, I and I plus 4, and the hydrogen bonding distance majorly found is 3.4 angstrom. Okay. Then in right-handed alpha helix, you find a full turn. A full turn includes 3.6 amino acid residues in one turn, and the pitch of that helix, like in one turn, the pitch will be, pitch means how much rise it is have. Total one turn, how much rise it has. So that is termed as the pitch. So if this is a full turn, then this rise will be 5.4 angstrom. Here from here to here, this is 5.4 angstrom, and total 3.6 amino acid residue will be involved in the one turn, complete one turn, and the rise is 1.5 angstrom per residue. That is in the commonly found in right-handed alpha helix. However, in 310 helix, these have the smaller pitch because they form and they also are tighter. So they form the hydrogen bond between I to I plus 3, and in pi helix you will find a larger pitch, which is the hydrogen bond forms between I to I plus 5. Okay, so the two types of beta strands are parallel and anti-parallel. The formation of alpha helix occur only when the continuous chain is present. This is very important point because alpha helix formation requires the continuous chain. However, in beta, the formation of beta strand, that may not be the continuous, because the function of beta sheets are to bring the two different two different part of the protein. They bring them closer by the formation between one part of amino acid, the amino acid present in the one part of protein, to the amino acid present in the another part of protein. Here the continuous chain is, if you see as a one, here the amino acid is 1 2 3 4 5 6 like that. So the bond formation, hydrogen bond formation will be from, if it ends here 7, then here 8, 8 might be in the loop, then 9 10 11 12 13 14, maybe here is 16. Then you will see the bond formation between the 3 and 16, 2 and 17, 4 and something else. So so these bond formations is in the beta pleated sheet, whether it is anti-parallel or parallel, they bring the two different part of the protein closer. Okay, so in the beta pleated sheet secondary structure, it may not be in the continuous fashion, but in the alpha helix formation, it must be in the continuous fashion. Okay, I hope this is clear. Now coming to the question, another question, which is which type of bond are the backbone of secondary protein structure? Read the question again. What type of bonds are the backbone of secondary protein structure? Options are protein bonds, option B amino acid bonds, option C hydrogen bonds, option D van der Waals interactions, option E amide bonds. Can someone answer?
Sir, option C, hydrogen bonding.
Hydrogen bonding. Very good. The correct option is hydrogen bond, because hydrogen bonds stabilize the interaction among the amide and carboxylic group of the main chain of the polypeptide. These interactions may include the formation of alpha helices and beta pleated sheets. So in secondary structure, normally these are stabilized by the hydrogen bonds, as you might have seen, and we were discussing from so long, and you have also watched the lectures, that hydrogen bonds play a very important role in stabilizing the secondary structure. So your another question is, why are anti-parallel beta sheets more stable than parallel beta sheets. So this is also give you an important point to remember that anti-parallel beta sheets are stable than the parallel beta sheets. The question is why? So can someone answer? Your options are the hydrogen bond angle is 150°. Option B, there are more hydrophobic interaction between the amino acids. Option C, the hydrogen bonding angle is optimized by anti-parallel sheets. Option D, the anti-parallel sheets are composed of more stable amino acid. Option E, the more covalent interaction between each amino acid. According to you, what can be the correct answer for this question?
Sir, option C, hydrogen bonding angle is optimized by the anti-parallel sheets.
Okay, and why do you think so?
Sir, because in anti-parallel sheets, one sheet is from right to left and other one is from left to right. So
Okay,
they are more closer, the carbonyl group and the nitrogen of the amine to form a hydrogen bonding. Whereas in
okay,
parallel sheet, the sequences are same. So that lead to the lesser stability in the parallel sheet as compared to anti-parallel.
very good. So your correct answer is option C. The hydrogen bonding angle is optimized by beta anti-parallel sheets. Explanation is in anti-parallel beta sheets, the hydrogen bonding angle is 180° and optimal. Okay. However, in the most stable, and this is the most stable angle, as you correctly pointed out, that in anti-parallel fashion, they are the more closer and it will be easier to form the hydrogen bond between the NH and CO. Okay. However, in parallel sheets, it is less stable, which is 150°. So whether a sheet is parallel or anti-parallel does not tell us anything about the amino acid it is composed of. So each of the other answer is incorrect because uh, these are there are hydrophobic interaction. This has nothing to do with the stability of the anti-parallel or parallel beta sheet. So the correct answer is C. Your question number 6 is in the contact map given here. In the contact map given here, what X, Y, and Z represents? Your options are X. Option A, X is anti-parallel beta sheet. This one. Y is alpha helix. Z is parallel beta sheet. Then X is parallel beta sheet. Y is alpha helix, and Z is anti-parallel beta sheet. Option C is X is alpha helix. Y is parallel beta sheet. Z is anti-parallel beta sheet. Option D is anti-parallel beta sheet, Y is parallel beta sheet, and Z is alpha helix. What is the correct answer for this question?
Sir, option A, X is
Option
anti-parallel,
A.
Y is
Mhm.
alpha helix, and Z is parallel sheet.
Okay, why do you think so? Can you explain?
So because in the lecture, ma'am has told that there is a diagonal from which we can plot a map. As a result of that, we can see whether the amino acids are in alpha helix or beta turn. So the diagonal is from the zero line. So the something
Mhm.
which is parallel to zero, that is Z. So it is a beta sheet. And X is anti-parallel to the diagonal, so it is anti-parallel. And Y is alpha helix because in alpha helix there is only a hydrogen bonding, and in beta sheets there are two different sequencing which are joined by a hydrogen bonding.
Okay, so your answer is X is anti-parallel beta sheet, Y is alpha helix, and Z is parallel beta sheet. Can you tell
Yes.
me why alpha helix falls in the diagonal? Why do you see only alpha helix arranged in a diagonal fashion? Why do you
Uh,
see that?
because as ma'am had told us that we have a ith amino acid and I plus 4 amino acid. So when we just plot a graph, so the amino acid which form a hydrogen bonding is with, like, say, the first amino acid form a hydrogen bonding with the fifth one, and the second one will
Okay.
form a sixth one. So when we do a mapping, so in a mapping there will be a straight line along the diagonal.
Okay. Very good. So correct answer is option A. Your next question is A sequence of amino acid in a certain protein is found to be serine, glycine, proline, glycine. The sequence is most probably part of A or N, antiparallel beta sheet, alpha helix, parallel beta sheet, or beta turn. What would be the answer for this question?
Sir,
Try to
option D.
understand the characteristic of amino acids that are given in the sequence. What did you say? Option?
The option D, beta turn,
Option D.
because
Oh, why?
as you told us that glycine and proline, they are the helix breaker. So
Okay.
they can't lead to the formation of alpha helix. So the beta helix chances are very much lesser. So that will be a beta turn or a loop one.
Okay, good. And also, you mostly found proline in the term because of its immuno group present in that. So they are formed. They are mostly present either in the starting of a alpha helix or either they are found in the term formation. So the correct option is beta term. Very good. Next question is a D amino acid would interrupt an alpha helix made of L amino acid. Another common naturally occurring constraint on the formation of alpha helix is the presence of option A, negatively charged arginine residue, option B, a positively charged lysine residue, option C, two glycine residue side by side, option D, a proline residue. We have discussed this question many times.
Sir, option D, proline residue.
Good. So the correct answer is option D, a proline residue. Question number 9. The most important contribution to the stability of protein conformation appears to be option A, sum of the free energy of the formation of many weak interactions between its polar amino acid residue and surrounding water. Option B, sum of free energies of the formation many weak interaction among the hundreds of amino acids in a protein. Option C, entropy increases from the reduction of ordered water molecule forming a solvent cell around it. Option D, maximum entropy increases from ionic interaction between the ionized amino acid in a protein. What do you think about this question? This is a long question, so you read it and then try to understand and answer it. The most important contribution to the stability of protein conformation appears to be. What is the correct answer for this question? What does it appear to you?
So it can
Yes.
be option C, entropy increase from the reduction in the order water molecules forming a solvent shell around it.
Okay, why do you think so?
So because in thermodynamics we have studied that the delta G Gibbs free energy must be negative. So similarly, the
Okay.
entropy must increase due to which the stability can be taken into account. So whenever we
Okay.
have a water form the hydration energy. So when we have a reduction of the ordered water molecule, it means they are in random chaos. As a result of that, entropy will increase.
Okay. Very good. Correct explanation, and your answer is also right. Option C is the correct option. Entropy increases from the reduction in ordered water molecules forming a solvent cell around it. Question is, which of the following amino acid is least likely to be found in the middle of an alpha helix? Again, the question is repeating though, so let me answer it. The question is, the answer is proline, right?
Proline.
Any doubt? No, no, because we have discussed this many times. So your next question is, in a sequence of amino acid or within the alpha helix, between which amino acid is the sequence does hydrogen binding occur? Every how many amino acid? We have discussed it already. So what is the correct answer?
Sir, option B, 1 and 5.
1 and 5. Are you sure?
So let me check it out. Let me just check it. So yes, because we have a I and I plus 4
Okay.
residue.
Yes.
So If I is 1, then I plus 4 is 1 plus 4, it 5, it makes 5.
So in an alpha helix, hydrogen bonding occurs every 4 amino acid residues starting from the first binding to the 4th in the sequence. Okay, so you will find the first binding to 1 and 4th on the sequence. So your correct answer is C, not B. Every 4 amino acid residue, right? Not 5, because it will make it as a I plus 5. Right? If your I is here, so every 4 amino acid residue, there will be a hydrogen bonding formation. So first will interact with 4th. I guess there is some problem. Okay, we'll check this. But the next question is the formation of dash is determined by dash interaction, whereas the formation of dash is strongly influenced by dash interaction. Your options are alpha helix, short range, beta strand, long range. Alpha helix, long range, beta strand, short range. Alpha helix, long range, alpha helix is short range. Beta strand, short range, beta strand, long range. What is the correct answer for this? Question number 10.
Sir, option A, alpha helix is determined by short range interaction and the formation of a beta strand is influenced by the long range interaction.
Okay, very good. Next question is, which property is shared by both myoglobin and hemoglobin? Option A, both are saturated with oxygen at low oxygen concentration. Option B, both display cooperative bonding when transporting oxygen. Option C, both contain the strands of beta pleated sheet with a zigzag shape. Option D, both contain large segment of alpha helix with a spiral shape. Which property is shared by both myoglobin and hemoglobin? What could be the correct answer for this question?
Sir, option D, both contain large segment of alpha helix with a spiral shape.
Why other options are wrong? As per
So
you?
because sir, in a hemoglobin there are 4 subunits, so the cooperativity is there in a hemoglobin. It is not in
Yes.
myoglobin.
Yes.
Moreover, by studying the hyperbolic and the sigmoidal curve for the myoglobin and hemoglobin respectively, so the A option is also not there. And the beta sheet with the zigzag shape, so it will be difficult for the hemoglobin or myoglobin to be accumulated in a particular blood cell. So that is why the option D must be there.
Good. Very nice. So correct answer is D. Question number 12. Determination of the precise spacing of atom within a large protein is possible only through the use of which technique? Option A, Ramachandran plot. Option B, molecular model building. Option C, X-ray diffraction. Option D, contact map.
Sir, option
You
D.
might have studied contact map. Okay. Any other options?
No, sir, no. Sir, the spacing is determined by the Bragg's equation, that is n lambda is equal to 2d sine theta. So it must be a X-ray diffraction. Sorry, sir, it must be X-ray diffraction.
Okay. Yeah. So your answer is correct. Ramachandran plot is nothing but the plot between phi and psi angle to know which are the allowed region and which are disallowed region. So option A is wrong. Molecular model building is also not a correct option because these are to predict the 3D structure or the functional form of protein from a linear protein sequence is given. So this is also not to determine the spacing. So as you rightly mentioned that Bragg's equation tells about the spacing between the atoms or the how the crystal structure or the different atoms are arranged in a fashion of the crystallinity or in all that is determined by the X-ray diffraction. And contact map, you all know that is the plotting of the distances or the type of helix that type of the secondary structure are present in the plot. That is the contact map, right? As we have discussed in the earlier questions. So correct answer is C, X-ray diffraction. Now, next question is which of the following statement about the oligomeric protein is incorrect? Option A subunit may be similar to other proteins. Option B, all subunit must be identical. Option C, some oligomeric proteins can further associate into large fiber. Option D, some subunit may have non-protein prosthetic groups. What is the correct answer according to you?
Sir, all subunit must be identical. This must be the incorrect one.
This must be an incorrect one.
Yes,
And
sir. Means the correct option. All subunits must be identical.
okay, good. So the answer is, where is my cursor? Yeah, here. So correct answer is B, because this is incorrect statement in terms of oligomeric protein. Now, question number 14 is which of the following is not true about secondary protein structure? Option A, the hydrophilic hydrophobic character of amino acid residue is important to secondary structure. Option B, the ability of peptide bonds to form the intramolecular hydrogen bonds is important to secondary structure. Option C, the alpha helix, beta pleated seed and beta turns are example of protein secondary structure. Option D, the steric influence of amino acid residue is important to secondary structure. What is the correct answer for question number 14?
Sir, option A, hydrophilic and hydrophobic character of amino acid is important to secondary structure.
Okay, very good. Because this character does not determine whether this will form a alpha helix or beta acid. Rather, the steric hindrance and all that determines whether, such as in glycine and proline, the structure information rather than the hydrophilic or hydrophobic character. So the correct answer is A. The hydrophilic and hydrophobic character of amino acid residue is important to secondary structure. This is the incorrect statement about the secondary protein structures. Now question number 15. Indicate which secondary structure or structures of the following peptide is likely to adopt in an aqueous solution of pH 7. Similar kind of question ma'am has also discussed in her lecture. So in your answer you also mention why what kind of criteria you need to check if you want to identify whether this is a which which structure it will follow. So uh, how will you approach this question? Question number 15.
Sir,
Anyone?
option B, beta plated sheet.
Beta pleated sheet. Why?
Because of the abundance of glycine and alanine group which are there in this 15 amino acid residue.
Okay. Yes. So the correct answer is beta pleated sheet because glycine presence of glycine and repeated glycine alanine both are the small amino acid residue. So uh, they may form the different kind of conformation, and because of it destabilizes the alpha helix, because this can have the different like conformation or arrangements due to its smaller in nature. So repeated glycine and alanine, if you find it in a protein sequence, then you can correctly identify this will probably form a beta pleated sheet. Okay, so your correct answer is B. Question number 16. Which of the following is not observed in a contact map? We have already discussed about the contact map in previous question. So try to answer this question. Option A is alpha helix appear as thick bands along the mean diagonal. Option B, parallel beta sheets appear as thin bands parallel to the mean diagonal. Option C, alpha helix appears as a thin band perpendicular to the mean diagonal. Option D, the antiparallel beta sheets appear as thin band anti-parallel to the mean diagonal. What is the correct answer for 16?
Sir, option C, alpha helix appear as a thin band perpendicular to the main diagonal.
Yes, because alpha helix normally appear in the diagonal, right? In the main diagonal, as we have discussed in the previous question. So the incorrect statement here is C. So correct answer is C. In an alpha helix, the R groups of the amino acid residue are found to be the outside of the helix spiral, generate the hydrogen bonds that form the alpha helix, stack within the interior of the alpha helix, or alternate between the outside and inside of the helix. What is the correct answer for this question?
Sir, option A, because they are found at the peripheral of the helix spiral.
Okay, correct. Because in the backbone, you find a peptide bond, and however, in the outside of the helix spiral, you find that R groups are protruded. Okay, so correct answer is A. So now this concludes the questions that were in the previous year assignment. So I have few more questions. Just this question was already answered. So which of the following amino acid is found in the beta turns? Which of this in beta turn?
Proline.
Correct. So sorry. Hmm. Next question is a dash is found in the antiparallel beta sheet where the first and last strands are connected by the hydrogen bonding. What could be the correct answer for this? This is related to the motif.
Sir, helix turn helix.
No. Here in the anti-parallel beta sheets, like in anti-parallel beta sheets, when the first and last strands are connected, they form a kind of beta barrel structure. And these structures you find normally in the porins, kind of when you in the membrane protein, when the porins kind of structure you see, those are normally found by the beta sheets, and they form a particular kind of structural form which is beta barrel. Okay, beta barrel, you can understand them by they form a kind of cylindrical structure, and they are formed by the beta sheets surrounding them. Okay, and they form kind of a helical. So these are called the beta barrel. So correct answer is C, beta barrel. Now, as I had said that I will be discussing about the research perspective of this particular protein architecture. Here, you have studied in your lectures, like in the module 2, that to know the sequence of polypeptide. If you isolate a protein, if you let us say if you want to find out what is the particular function of a protein, or if certain role is being played and you want to understand which protein is in behind it, so first thing you would like to know that what are the sequence involved in that protein. If you know the sequence, then you can understand that by how this can be folded, because to understand the folding of a protein, you also need to know what all the amino acids are present. Okay, so what you do, you isolate the protein, and then you, by the techniques such as the degradation techniques, Edward degradation methods, and other methods that ma'am has already discussed, you first identify which all amino acids are present in that, and then what you do, then you go for the mass spectroscopic technique. So here, uh, if you understand the structure, if you want to understand the structural biology, how the fashion goes in the understanding a protein structure, you first isolate the protein, then digest it. This was the previous step that you saw in the previous slide, digestion, and then you find a peptide mixture. This digestion can be of the small peptides, or also at the different amino acid level also, by the different advanced degradation method. In that you first the reagent attack to its the first carbon, like first amino acid, then it that is being degraded, and then the second, third, sequentially you identify the different amino acid, and then you alternative to it, you can digest it with using the protein digester molecules such as protease. So that will give you a small peptide mixture, and then you identify them by the multi-dimensional chromatography. It can be the like any kind of chromatography, column chromatography, then HPLC, GCMS. Likewise, you understand what all amino acids are present in that. Okay, so these all techniques, if you have not heard about it, no problem. Ma'am will discuss in the later lecture to identify how the protein, which all the proteins are present in a mixture. Then you need to run the chromatography techniques. You can understand them by the chromatography technique. And when you take that structure, those samples, you get in the different tube, and then you subject it to the tandem mass spectroscopy or mass spectrometric technique. Then you get a sequence like the uh, sequence of amino acid, like different amino acids present, and then similarly you find out how, then you get a probable chance of the amino acid sequence. Then you match this with a database that are present, the library of the mass spectroscopy, and then you identify the amino acid sequence. Then you run the blast in the computational technique, and you identify which amino acid, which protein it is, and that's how you identify the protein sequence. This is also the similar thing explained in a different way. Here the same thing, result report, you find out which are the amino acid present in them, and then like by searching in the blast, by blasting it to the database of the protein and DNA sequence, you find out which peptide bond is that, and you identify that particular protein if that is unknown. Now, the another technique, this is like bonus point I would like to give you. This technique is known as the drug and affinity responsive target stability. So here also you take the knowledge of the protein architecture here. What the main objective here to identify in this as the name says, drug affinity responsive target stability. This is kind of a experiment where you identify a protein which is interacting to a particular kind of small molecules such as rampamycin or FK506 or any inhibitor that you want to analyze, or you want to analyze certain kind of mycotoxins. So in that, what you do, you prepare a cell lysate and you add a drug to it. Okay, so if the principle behind the darts is if the drug is binded to that particular protein, that will stabilize that will hinder the protein cleavage site. So that the proteolysis enzyme, like enzymes that degrades the protein, does not degrade, cannot degrade this particular kind of protein in which the drug is bound. So the rest protein which is not bound to the drug, that will be degraded. So like here, in the cell lysate, here no drug was added. So when you proteolyze this, then this will this protein will be degraded, and you, if the drug is attached to it, this particular protein will not degrade, and you will find a drug target of a particular protein. Then what you do, you take this sample, and then you put it like you take both of the sample and you put it in the SDS page. You run the SDS page analysis, and then you identify, if you find a strong band, which means that protein is not degraded, then you cut it and subject it for the mass spectrometry, and you identify that particular protein, so that you understand, or what is the target of that particular drug, and particular drug, and which kind of, which drug, which protein does it the drug bind to. Okay, so this is how you understand the protein ligand interaction using darts technique. Okay, so next thing is here, how the XRD works. So these two topics will be discussed by ma'am, but I would like to give you a brief understanding about it. As Karan already mentioned that the in the Bragg's law, like in XRD, if you put a single crystal in between the lead screen and the photographic plate, this crystal is being rotated and you put a X-ray beam, you bombard X-ray beam onto the structure. Then the diffraction happens due to the electrons present in their outer surface, and then they diffract the light. Okay, they diffract this X-ray beam, and this is taken on a photographic plate, and you find the arrangements of the atoms. So likewise, you understand the spacing and you map the structure of that particular crystalline compound. Another technique here, this graph is representing the circular dichroism chromatograph. Circular dichroism. So circular dichroism is nothing but a kind of absorption technique where you put a light source and which is unpolarized light. Then you pass this unpolarized light to a monochromator. What this monochromator does, it converts this unpolarized light to linearly polarized light, which is also a polarizer. Okay, so these are the linearly polarized light. Then you pass it through a kind of, say, a film which converts this linearly polarized light to circular polarized light, which may be of the right circular polarized light and left hand circular polarized light. So if these polarized light are subjected to the sample, then the samples absorb this light in different manner, and by that the absorptivity, then you calculate using the absorption law. Then you calculate the difference of the molar absorptivity of delta epsilon of the absorption light of the right circularly polarized light, the difference between this right circularly polarized light and left circularly polarized light, and then you find out what is the absorption, and then you identify which kind of structure are present in that particular sample. So here you see that this is the change in the epsilon, and you scan through the wavelength of in the far UV range. Okay, to not far UV range. This is near UV range, 200 to 250 nanometer. So in that range, you understand which kind of sample is present. If the sample is present in like that, so this red graph is for the alpha helix. In alpha helix, you find a strong band at 205 nanometer and 220 nanometer. However, this is a beta sheet. Black one is the beta sheet. So likewise, you identify which kind of secondary structure is present in that particular compound, uh, particular sample. So this concludes the session on the second week. So thank you all for attending this session. So I hope you all have understood the concept behind the protein architecture and some of the topics. The if you These two topic will be also taken by the ma'am in the upcoming lectures, but I just gave you the brief because in some of the question that appeared the circular dichroism and also about the XRD. So that is why I discussed this. So this is my information about it, and the references are listed here. You can go through these references, and thank you everyone for attending this live session. So I will be uploading this recording on the YouTube channel. And if you have any doubt, you can ask me, or you can also post your question in the discussion forum. Some of any of the TA that are listed in this course, they will answer, or you can also direct an email to me on my email ID karan18@iitg.ac.in. I have also created a Telegram channel. You can join this Telegram channel, and if you have any question, you can interact me there also. Thank you so much for your patience listening. If you have any question, you can ask me. I am happy to answer any question related to this. Or you want to give a feedback, you can also give a feedback to me. Did you find any difficulty while going through this lecture?
No, sir. Very well explained by you, sir. Thank you so much.
Thank you, Karan. Okay, anyone else want to give their feedback, and also if they want to ask some question related to this module too? Okay, thank you for joining. So I will stop the recording.